Mhm. Freya, you hear us and you see the screen? Okay, very good. Uh, Good morning to everybody. I see so many people today. Well, I appreciate that you after such a long year end of the semester that you really appreciate that you coming. Thank you. And we continue with our course, and today we will briefly return to the previous topic. Somebody maybe have mouse. Yeah. Okay, that will be much better because I forget in my office. I don't like very much this touch mouse and the. I have a whole bunch of them in my lab. Okay, very good. Thank you. Don't forget to take it from me.
That will work, so maybe press and move something.
It works. Yeah. Oh, much better. We will. It was some inaccuracies from my side last class when I spoke about neurophysiological effect of opioid ligands. We will briefly come back to this topic. And so I showed you our studies with a selective DOR agonist SNC80 and selective DOR antagonist anti-naltrindol, which in the future slides it will be shortened for NTI. And what we did first, we checked acute effects on hippocampal glutamate and brainstem monoamine neurons. On hippocampal neurons, when we applied first SNC80, it was suppressive inhibitory effect, and subsequent administration of naltrindol did not reverse it. But we first applied naltrindol antagonist, then agonist could not affect the firing rate. With 5-HT neurons, we also make two options. In the first experiment, we applied first agonist, and we saw decrease, and with antagonist, we did not see any further effect. But when we applied first antagonist, we saw increase, and agonist recovered this increase back to the baseline. And with noradrenaline neurons, we did not see any statistical significance. We have some tendency here, but it was not statistically significant. And with we saw statistically significant effect only with a combination of naltrindol and SNC80, and only when SNC80 was administered first. Probably, if you can see this paper, our conclusion was that the effect of glutamate, serotonin, and dopamine neurons is indeed DOR delta opioid receptor mediated. And this effect probably noradrenergic, probably not DOR mediated, because SNC80 is known also to affect directly sodium channels. A chronic experiment we did only with glutamate, dopamine, and serotonin. With serotonin, we did not see any effect. With glutamate, we see increasing effect. Same with dopamine, we saw increasing stimulatory effect, opposite from what we saw in the acute experiment. And this was our hypothesis how to explain it. That acute SNC80 increase extracellular 5-HT, which suppress 5-HT glutamine and dopamine neurons. And after 2 weeks, we have sensitization of serotonergic receptors, and only then we see only direct opioid mediated effect. Okay, any questions to naltrindol? Now I hope it's clear. Yeah. Then we will go to today's topics. It's interaction between corticosteroids and monoamines. I really like this topic. Unfortunately, I stopped for a last time. I stopped working on it from all kind of reasons. But I really hope to go back because I think it's very, very interesting, very promising topic. We all we spoke about depression now before mostly, but depression is only one of so-called stress-related disorders. It's disorders which stress in some degree involved in their etiology. Other stress-related disorders, it's a GAD, generalized anxiety disorder, post-traumatic stress disorder, panic disorder, and obsessive compulsive disorder. We experience stressors from all through our life. We start to experience stressors when we still we started to experience stressors when we were still in uterus of our mothers. For example, maternal immune activation, acute infection, maternal illness, we will see in the future, soon we will see. This is a pretty strong stressors. Juvenile stressor can be abuse, neglect, or much less aggressive form, for example, parental separation, which unfortunately happens pretty frequently, and this is indeed still stressor. And we experience many stressors as adults, and one of the most severe stressors as adults when we experience or observe life-threatening situation. We should not see the wrong way to see stress. First of all, we need to be very clear what is a stressor and stress. Stress, my collaborator Daniela Yezhova in Slovakia, she is a very competent, very experienced stress researcher. He hates expression stress response, because there is no stress response. Stress is already response. A stressor, it's any kind of event, factor, chemical, physical, biological, within which possess challenge for our homeostasis. And stress is a response to this factor. And stress should not be really negative. There are many positive stressors. Sometimes we search for stressors. You see here, it's less common in Israel, but way of recreation, but very common in Europe, especially in North Europe, sauna. Can try to imagine sauna. It's first of all, you put your organism to the temperature of plus 100, and then you put it to plus 4. So suddenly you put your body to the condition, you set your body signal to the condition when you cannot survive more than 10-15 minutes. It's the extreme stressor, but we like it, people do it, and according to the most of the opinions, reasonable usage of sauna is very good to the body, actually. I'm coming from Slovakia. In Slovakia, it's a home. In today's Slovakia, in Komarno, was born Hans Selye. Hans Selye, he was considered the pioneer of stress research, and he distinguished between eustress and distress. Eustress is a positive stress, like what we speak about sauna, and distress is a negative stress which leading to logical problems in the body. And many researchers and my lab between them try to understand what's actually the difference. It's maybe the very crucial factor to understand why and which point we go to eustress, from eustress to distress. Probably the answer we should see in the crosstalk between biomolecules which mediate stress, response to the stressors. And most of them we already met during this course is serotonin as a mediator of anxiety is very important in the stress. Each time I want to say stress response. I try not to tell it because it's a wrong expression, but we're so used to tell it. And catecholamines, dopamine and adrenaline. And catecholamines, they act in the central nervous system, but also in the periphery. Today we will go more from the brain to periphery. We will see that periphery, those peripheral organs, they extremely important. And central nervous system, we have mostly noradrenaline. In adrenal gland, the common belief that adrenal gland, we have only adrenaline, but it's not true. Adrenal gland, middle of the adrenal gland, release both adrenaline and noradrenaline, and also a little bit dopamine. But dopamine, it's not good when adrenal gland start to release more dopamine as a sign of malignant tumor of the adrenal gland. And but adrenaline and noradrenaline, they both released, and there is some theories that noradrenaline and adrenaline, they also the proportion between adrenaline and noradrenaline, it somehow corresponds to the type of the stress response. If the fight response to the stressor. If the fight response or flight response. For example, predator animals like big cats, they adrenal release mostly 100% noradrenaline. And cows, they have 100%, almost 100% of adrenaline. Primates, including humans, we have around 50-50. And there are some studies that it also depends on your professional personality and what you're doing with your life. There are some studies that say combat soldiers, they have much more noradrenaline than adrenaline. And common men, they have a bit more adrenaline. And another type of biomolecules, very important biomolecules released by adrenal glands, we which mostly spoke about them today, it's corticosteroids. Corticosteroids, they call corticosteroids because they from cortex of the steroid molecule from the cortex of adrenal gland. And we have cortisol in humans. In rats, it's instead of cortisol, rats, they have corticosterone. And it's a glucocorticoid. And we also have mineralocorticoids, which are less studied, but they're not less important. It's the most common mineralocorticoid in humans and in primates and in rodents, it's aldosterone. I will show you two studies, two models of the stress and the response to the stressors in animals. And we will see how corticosteroids and monoamines co-work differently in these two types of stress. And from this, maybe we will try to identify the very basic pathways which separate stress and eustress. It's a very common model, pretty used model. Maybe some of you heard about it, the prenatal immune challenge. Somebody heard about it? Somebody you worked with LPS, lipopolysaccharide? Lipopolysaccharide, it's a component of cell wall of gram-negative bacteria. And if you can take this LPS and you inject it to the animal, animals start immediately develop immune response like to the infection, but it's not actually infection. There is no bacteria is not present in the body, bacteria is not multiplying in the body, but our organism, our organism identify it as infection to make more correct, we will see it later. And develop its activate cascade of cytokines. It induces seeking behavior. Animal feels sick, like it would be experiencing infectious illness, but it is not actually illness. There are several studies what will happen if we will apply this LPS to the mother during the pregnancy. And the effects are pretty strong. Actually, maternal maternal immune activation, it's a kind of model for the depression. We see pretty strong anxiety in the offspring of the mothers which experienced immune activation. You see depressive-like behavior, increased immobility during the fourth swim test. You see anhedonia. And there are also pretty strong neurochemical alterations. There is a decreased expression of BDNF. And there is decreased neurogenesis in these animals. Well, I don't want to make everybody scary, because most of, I believe, our mother, or more likely our mothers, they could have some acute illness during pregnancy. Some of you who were pregnant have kids may experience it. It's not that it's necessary. We'll do something very bad, but still this factor, this stress is underestimated and not well studied, because from animal studies we do see that it has pretty strong, pretty serious effect, and especially no one still know how this COVID-19 pandemic will affect the next generation in the terms of the mental health. And what unfortunately we cannot do nothing about it. It's happened and it will have some effect, but we need to study this, and we need to get prepared to negative effect which it may have. But I feel that the governments and intergovernmental organization grant and bodies which provide research grants, they're not very pretty. They're not even enough serious about this topic. ILPS is bacterial. There is similar. We used LPS because it was established protocol in the laboratory of my partners in Slovakia. There is poly-AC, which is a model for viral infection. In terms of effect of neurobehavioral effects, they are pretty similar. There are some difference, but they are more. If you compare different studies with prenatal LPS or prenatal poly-AC, more or less you find similar results. But COVID is much more serious, because COVID is much higher activator of cytokine system. But what was very bad with the COVID-19, we cannot we don't have rodent models. This virus is not affecting rodents. So this is a big challenge. We don't know how to investigate it. Then probably we still use some models via this model that we have. Now we go to our experiment. It's how we did experiment. We first we put male and female rats together, and we cannot really see the. Sometimes it happens that you see it, but rats are more active at night. They also copulate, they also perform sexual activity also during the night time. So how you can identify the beginning of the pregnancy? We have to get vaginal smears, take vaginal smears to the females every morning. You should do it very early, to come at 7 o'clock, because with the time sperm cells the vaginal extern they cytotoxic, that sperm cells they disappear within 2-3 hours. And when you detect sperm cells in the female vagina, it's almost 90-95% that she's pregnant. In the rodent's pregnancy, almost automatically after copulation. Then we wait. Then during this 15 to 19th of the gestation, you should be careful in English. Pregnancy in English, what I learned, I submitted paper and we had asked to correct it. Pregnancy is only, in English, it's terms related only to humans. In other animals, it's gestation. I did not know it myself. I submitted paper and I wrote pregnant rats, and I was asked by one reviewer, asked me to correct it. At day 21, it's usually the end of the gestation and the birth. And during the first 7 days, we examined maternal care behavior. I will explain you later what is this and why we did it. And then we left rats alone till the adulthood. And when they were 2-3 months old, we performed in vivo electrophysiology assessments. What is the maternal care behavior? We checked several times per day, such a paragraph like nursing, being nest, leaking nurset, blanket nursing, and nest building. There are two types of nursing in rats. In one type, the rats sitting on half, how to say it, a little bit with half-raised legs to allow to pups very easy access to the nips, and another it's a rat like lying how is good for her, and pups need to find access themselves. And why you think we need to analyze this maternal care behavior? Who can tell me? Yes. Exactly, because animals, because this is model of the sickness. If you don't check maternal care behavior, the answer can be you found some abnormalities. It's not because of maybe it's not because of maternal immune activation, maybe because animal felt sick. Because animal felt sick, it's less to care about pups. She took care about pups. I still don't know what. I think today in English you need to speak he and she about animals if you know gender. Once in classical English, animal was it. Well, if somebody say about my cats, it. Let's see about. But here we found some very interesting. It was not only it was no impaired maternal care behavior. One parameter, leaking nursing, was even higher in rats which experience sickness. We don't know really how to explain it, but it was even maternal care than in control rats. And then we checked electrophysiology and we found something very interesting. We found decrease. We found that in male and female offspring of LPS treated of prenatally LPS treated rats, IPC neuronal firing activity is decreased. But if you take only males and not females, we have increase in dopamine neuronal firing activity. And we published a study, and our suggestion was that LTS responsible to that decreased the 5-HT neuronal firing activity is probably linked with depressive-like behavior and with impaired neurogenesis and neuroplasticity. And increased dopamine, maybe it's a compensatory mechanism. And we saw before that serotonin and dopamine, they negatively linked, that serotonin decrease dopamine neuronal firing activity and vice versa. Maybe males, we are now running experiments to check it. Maybe males, they have more strong interaction between serotonin and dopamine system. And when you have decrease in serotonergic system, you have compensatory increase in dopamine, which may correct, at least partially, some of the problems caused by decreased serotonin. And maybe it's also one of the factors which can explain why we have higher prevalence of depression in females. Maybe it's still hypothesis, very far from to be confirmed, but what it says really very important to include different sexes when in all stress-related research, because yes.
Like, how sure are you in this result? Is something that, like, when you get a result, something that you don't expect, to say, okay, I see this is significant, but you need as a scientist to like to feel it, right? It's more of a personal question. How can you redo this experiment a couple of times to say, okay, now I have this very, very big elaborated theory on top of these results?
Well, decrease in serotonin we expect.
Dopamine compensation, maybe.
I'm not sure if it's compensation right now. I'm not very surprised why we see it here, because we saw a negative, we saw DV-link, we saw that it was Bruno Gouillard, my friend and co-worker during postdoc. He did serotonergic denervation and he got exactly the same increase in dopamine. So it's okay, it's more or less expected. Only in males, this is a problem. Till very recently, all research was done only in males. Because maybe 10 years ago, like 90-95% of the research was done in males. And it was considered to be, you should not use female rats in research because you're not sure about the uteral cycle. And it can be totally different results during estrus and during pre-estrus or post-estrus. And very few groups which worked with females maybe 10 years ago, they did ovarectomy, which is also not answer. Then it was when we started this research, it was very important to check that when you do electrophysiology, any experiment, to check that animal not in estrus, and to last time reviewers become much more reluctant about it. Now you just include both genders, and you don't check uteral cycle usually, which is also, well, there is no perfect answer. If you do everyday check the uteral cycle, is also additional. I'm not sure that rats very happy about it, that you touch them and it's additional stressor, of course. And if you don't do it, you do. Well, females, they do have uteral cycle, all females, and this definitely have effect on central limbic and monoamine neurotransmission. But definitely and this is a fact. We have we not ever repeated it. I don't know if any other group repeated it. We're now repeating it. We're now doing some more advanced study, and we go to repeat it. And we go to see if indeed females, they don't have this dopaminergic increase. But right now, according to what I see, maybe this interaction between serotonin and dopamine, and till now it was checked only in males. Maybe it's this interaction less strong in females. Yeah, we have plenty of studies, and no, not plenty. Now we have many other prenatal studies, prenatal antidepressant, with pregestational chronic and predictable stress. I could not include everything to this lecture. And you always see in everything related to the stress, and especially pregestational prenatal stress, you have very different response in males and females. Okay. And now go to another study, and we will be surprised to see pretty different effect. It's the effect of prenatal predator scent. I believe everybody heard about this model of predator scent stressor. Who heard about it? This model is Holavan to Seritaris. It was developed by Hadid Cohen. You heard about Hadid Cohen? You know, she is a pretty known and successful stress researcher from Beersheba, from Ben Gurion University. And she was one of the first developers of the models, maybe 30, 40 years ago. In the beginning, one of the model of the rat was to expose rat to the cat. It was usually the studies was that no, you don't allow to cat to attack the rat, but you put them together, then you take cat away, and but there is a problem in this model. Actually, healthy cat, he almost does not have smell, because cats they ambush predator. They usually attack from ambush, and their main prey rodents. Rodents very sensitive to the smell, and it's why somebody of cat have experience cat, why cats such compulsory always clean themselves, because if you don't clean enough and you have smell, then you lost your dinner. It's a very in the nature is very simple answer. And domestic cats, they don't need it usually to pray for food, but they still keep it. But what actually has even more strong effect on the rodents than cats, than real cat, it's a cat urine smell. Hadid Cohen, he built different models based on this. Is very differently if you expose the animal within the home cage, outside of the home cage, but the bottom line that exposure to the cat urine or it's very hard to collect liquid cat urine. Usually you just use how it's called, not sand. You usually use filament from the cat latrine. Some of you have cats. Well, yeah. In my case, in our experiments, I just took it from my cat's litter box. And rats, they definitely don't like it. Rats don't need to learn it. The rats don't need to be educated by mothers that if you feel the smell, run away. It's worked on biochemical level. For rodents, the smell of the predator is very aversive. It's a signal of immediate danger to the life and to the body integrity, and it's immediately it's immediate and pretty strong stressor. What we did in our experiment, Bitcoin usually her model is based on the single exposed to the predator stand. She exposed red ones and measured the behavior or neurochemical effect 10 days later. We followed, this time I collaborated with a Russian laboratory from Chelyabinsk, from Professor Seligman from Chelyabinsk, and he developed a modified model of chronic response. He claimed, well, it's I'm not sure how much it's true, that single response, it's a model for regular stress and prolonged repeated response to the stress or to the predator. So for 10 days, it can be model of the complex or combat related PTSD. After this rat were left alone, they rested for 2 weeks. And then we performed behavioral and electrophysiological study. First of all, we tried to check if rats really avoid, if rats really have, this is a real video from these cages. This is a plate, and the plate we have filament from the used cat litter box. And control animals, they have the same filament, but liquid, made liquid with clean water. Just see, it should be work. No, it's not video. It's audio. It's only picture. We saw that they really try to avoid it. That it was response. They really care about it. When we compared it with the liquid water, the rats try to avoid. The rats definitely try to avoid it. But then we made. Excuse me.
I think you always put it in a very tight box. Because mice really go to the corners anyway, so they'll avoid the center either way.
Anyway, but when you had urine, they avoided more than if it will be with the clean water. And we checked elevate EPM behavior after 2 weeks, and we were very surprised to find no anxiogenic effect. It was really surprising because I did coin. So where is after even single exposure and 2 weeks later, she showed strong anxiogenic effect. And Seligman, when he used it in his home laboratory after prolonged 2 weeks, 10 days exposure, he also saw anxiogenic effect. And then we probably figure out why we did not see it. He did it in the home cages. He brought this urine, this smell factor to the home cage of the rat every day for 10 days. We also wanted to copy his protocol, but our management of animal facility did not allow it because air conditioning system is common, and we would bring it to the animal keeping room. The smell could spread to other rooms, and that could affect all animals about facility. In Russia, probably they less care about such a thing, because maybe we believe that when you bring the stressor to the home cage, it's much more strong stressor. Maybe rats just learned after 10 days that they take them out and nothing happened. We were laughing about this. We wanted to make model of PTSD, but what we did finally, maybe it's a model of CBT, cognitive behavior therapy. I don't try to claim, I'm saying it very carefully, because maybe it's a CBT model. What we found, yes.
Not in their home cage.
Not in home cage. We took them every day. We took them to laboratory. We put them to these special exposition cages for 10 minutes, and we brought them back to the home cages. Maybe this was a critical factor, which made all this difference. And What happens when we check serotonin, when we check serotonergic neurons? You see, same direction, we did not see anxiolytic effect, and we saw increase in serotonergic neuronal firing activity, which is probably also compensatory mechanism. That rats, maybe this indeed some kind of CBT, and maybe CBT indeed works via increase of serotonin. When when a person re-exposed to the stressors and learn to copy with them, maybe it's happening in the way of increase in the firing of excitability of serotonergic neurons. Now we try to understand why these effects of prenatal immune challenge and postnatal prenatal stress opposite on serotonergic neurons. Somebody has idea why it can be the opposite effect? I will give the hint that perhaps interaction between monoamines and corticosteroids. Let's go to a little bit of corticosteroid signaling. You know that corticosteroids, they activate glucocorticoid receptors, GRs, unlike receptor which we saw before, ionotropic receptor and ionotropic metabotropic receptor. Glucocorticoid receptor, they mostly situated, gluco and mineralocorticoid receptor, they mostly situated within the cell, not on the surrounding the cell membrane, and this is important. But most of them, they within the cell. Corticosteroid is a steroid molecule. It easily penetrates the membrane bilayer. It activates intracellular receptors, and this intracellular receptor, after the activation, they don't need second messenger. They start to act as translation factors. They build up to the specific places on the DNA, on the chromosome, and they activate or deactivate expression of the specific genes. But there are some G protein receptors which are expressed on the cell membranes, and they interact with the G protein couplet receptors, such as 5-HT1A receptor, serotonergic receptor. And in this case, they indeed alters the second messenger pathway. Was found pretty long ago. This is a study from 2012 that corticosterone decrease the excitatory postsynaptic currents in serotonergic neurons. This study was done in, it's not in vivo, the study was done in brain slices. And they also found that in the glucocorticoid effect on serotonergic neurons involved 5-HT1A autoreceptor, glutamate receptor, and were very interesting endocannabinoids. And endocannabinoid receptor also involved in communication between glucocorticoids and serotonin. We are now performing this very initial study from my laboratory. These results I get when I was here. After I already left for Israel to teach this course. We use, we checked effect of dexamethasone. Dexamethasone, it's a synthetic steroid and agonist of the glucocorticoid receptors. Now you can find, Sigma Aldrich, they provide dexamethasone attached to nanoparticles, which are very fast bring it to the brain. And we can check the real-time effect on serotonergic neurons, and we also found the same results, that it does dependently decrease the firing of serotonergic neurons. What about prenatal LPS and glucocorticoids? There is a study of Lin and Van from 2012, and what they found is that rats that were prenatally exposed to the LPS, they don't have increase in basal corticosterone, but they have more strong response to the stressors. If you expose this rat to the stressor, it has much stronger corticosterone response than the control animals. It means that because they might have higher corticosterone tone, corticosteroid signaling, which may suppress serotonergic neuronal firing activity and lead to anxiety and depressive-like behavior. And what about predators and stress? We analyzed corticosterone levels and corticosterone and aldosterone levels, and we're very surprised to find that there is a decrease in both of them. And what is also very interesting, this decrease was in the circulating blood, but not in the tissue of the adrenal gland. If you don't, what does it mean? We did not check, unfortunately, until now, we did not check it directly. If you have same levels, how blood plasma levels of corticosteroid determined? They determined by two factors. One factor is biosynthesis in the adrenal gland. The second factor is metabolism. With metabolism mostly happens in the liver by cytochrome P450. If you don't have change in the adrenal gland, but you have less in the plasma, it means that probably metabolism is a critical factor playing a role. And we indeed tried to check it in another study. In this study, we inhibited the cytochrome P450. After inhibition of the cytochrome P450, we get suppression of the firing activity of serotonergic neurons. What happened with other, if we suppress metabolism of corticosterone, what happened with other neurons? With noradrenergic neurons, we get increased in the burst activity. And with dopaminergic neurons, we get, with both of them, we get some increase in the burst activity. And this was our suggestion how it works. It means corticosterone release from the adrenal gland and metabolized mostly by cytochrome, mostly in the liver, but some also in the brain. If you block metabolism, you have increasingly in corticosteroids, which via mechanism involving 5-HT1A receptor suppress serotonergic neurons. And because serotonin inhibits serotonin, noradrenaline, and dopamine, probably we get increase in, following the decrease in serotonergic activity, we get increase in dopaminergic and noradrenergic ones. Adaptive change after chronic predators and stress might be opposite. We have other vice versa. We have increase in metabolism. Because increase in metabolism, we have decrease in the circulating corticosteroids, and then we have increase in serotonergic neuronal firing activity. What is very interesting to tell, this interaction is reciprocal between cytochrome and monoamines. It's reciprocal, not only when you change this factor, cytochrome activity, you have effect of on the noradrenergic, on monoaminergic neurotransmission. The vice versa is also true. This is a study from the group from Poland of Professor Daniel, and they found something also very interesting. If you increase serotonergic neurotransmission, they just inject a precursor of serotonin to the lateral ventriculi. they have decrease in the expression of the different types of cytochrome in the liver. Same with noradrenaline. They increased. No, with noradrenaline is vice versa. They did noradrenergic denervation. They destroyed noradrenergic system. They also get decrease in expression and activity of the liver cytochrome. With the dopamine also known to be involved. If you activate D2 receptor, you get also decrease in the cytochrome expression. And who is interesting to investigate, to learn more about this topic, Seligman and me, we wrote review in 2020, which we tried to summarize so-called brain-liver circuits. What I want to summarize here, it's some mistakes which we are doing, that we sometimes too focusing on the brain by itself. But brain is brain response to the stressor not alone, but in co-working with other organs. Such livers definitely need to be taken into consideration. Also, when you design new drugs, because the liver is also same cytochrome, it's also main metabolizer of the for example, antidepressant drugs. Antidepressant drugs, from one side, they are meant monoaminergic neurotransmission, which immediately meant cytochrome activity, which in turn also influence the metabolism of this drug. And we still don't know how to really to solve these very complicated circles. Probably IT models should be used to in the future to. We tried some, but probably what's the future should be used when you think about treatment strategies and drug drug design. Yes.
Patients with antidepressant not going to avoid any substances which are normally broken down by cytochrome.
I'm not clinician, but my impression it's not enough. It's not enough attention put to this. For example, we spoke about benzodiazepines. Benzodiazepines, the enormous activator of cytochrome. Under benzodiazepines, cytochrome metabolize the benzodiazepine and all other molecules much more faster. Before clinicians understood it, it's led to several tragedies. Because people, for example, people with heart diseases after how is Centur in English? Centur, you know what I mean? Okay, because we have Freya, we need to explain. Well, sorry. And people in hospitals who were treated with anticoagulants, it was very common, almost an enormous number of hospitalized people, they received benzodiazepines. Because the teams, nurses, doctors, let them at least at night less us to sleep. They will sleep and let us to rest a little bit. And it's very common to describe benzodiazepines to hospitalized people. And some people who were treated for all kinds of coronal diseases with anticoagulants, and they also received benzodiazepines. How clinicians in the department, how they see how many anticoagulants you should take. They took everyday blood tests, they see level of platelets, and they adapt level of anticoagulants. And level of anticoagulants were adapted according to the metabolism enhanced by benzodiazepines. people get released home, stop benzodiazepines, and sometimes get hemorrhagic strokes because the dose of the dose of anticoagulant was much higher than needed in lieu of benzos. You understand me? Yeah? Same with antidepressants. Really, I think this is a topic which is not sufficiently addressed. Okay, we have time less, and we can start another topic, which I consider to be additional topic, is a nitric oxide monoamine interactions. Everybody heard about nitric oxide, I believe. Nitric oxide is a very simple molecule. It's a two-atomic molecule, NO. It's in natural conditions, it's gas without smell, without taste, without some kind of clear immediate effect on biological organisms. Is because of N2, gas N2 is very not chemically active. It's not easy to get oxidated. There are not that many in atmosphere. It's almost not present in atmosphere NO, but it does present in the biological organisms, in the biological cells, which play many important biological roles. Some like platelets activation, neurotransmission, inflammatory response, and vasodilatation. How we get nitric oxide in our organism? It's synthesized from arginine by special enzyme NOS, nitric oxide synthase. Nitric oxide synthase just for just catalyzed oxidation of arginine with and as a result we get nitric oxide. There are three types of nitric oxide synthase, NOS. The eNOS, endothelial NOS, it's present in the vascular system. iNOS, inducible NOS, present in macrophages. And neuronal NOS and NOS we found in the neurons and glial cells. Endothelial NOS is very important in the coordination of the tonus of the blood vessels. In response to the pressure, to the stress of the blood, it's induced vasodilatation via potassium-mediated mechanisms. But we are more interested in the nNOS. And nNOS first when nNOS in neural cells was discovered, its function was discovered, it was very amazing discovery, because what was found that it's act as a retrograde neurotransmitter. This is glutamatergic synapse. Glutamate get released from the presynaptic neurons, activate NMDA receptor. NMDA receptor via cascade of the second messenger activate nNOS, and nitric oxide leaks back to the presynaptic neurons and make a positive feedback, stimulate release of glutamate. It's not only retrograde, it's also anterograde neurotransmitter in synaptic cell. The nitric oxide is known to induce growing and transformation of the dendritic spines. I believe everybody heard about dendritic spines. I think last class we will speak a little bit about them. Chromatin is a simple molecule which close to arginine, is a derivative of arginine, and it acts as an inhibitor of all three types of nitric oxide synthase. It was discovered by German chemist Albrecht Kozel almost more than 100 years ago. And this molecule, they have also plenty of biological effect, but still now it's not this molecule, not its derivative used in the clinics, but it might have very interesting effect. For example, it was found that it has antidepressant-like effect, but it's not antidepressant-like. It did not have antidepressant-like effect on regular control animals, but it had antidepressant and anxiolytic-like effect on animals which experienced prenatal alcohol, prenatal alcohol exposure. It's a shame that that Shelley, I forgot your name, Shelley, she's working on alcohol. Well, it would be interesting to. It's also model of depression. If you expose rats prenatally to alcohol, distance they have depressive-like behavior, and this depressive-like behavior is corrected by agmatine. It was also another study. I don't show you results of this study. Some of you who are interested can read it. It's very heavy, very complicated study. They found that there are two types of serotonergic neurons in the raphe nucleus. Some of these neurons they express NOS, some don't. And serotonergic neurons that express NOS, they have less excitable. They have less firing activity. It means that NOS may have inhibitory effect on the firing activity serotonergic neurons. And we checked it. It was a very interesting story how I came to this. I never planned to work on NOS. I heard about nitric oxide, but I never had planned to work on it. And 1 year ago, it's in August, it will be 1 year. No, it's now already 1 year. One student from another institute came to me and told me that she wants to change supervisor for several reasons. That if she does not change supervisor, she has to quit to finish PhD studies. I noticed that she is a good student and let's give her a chance. And in her previous laboratory, she worked on nitric oxide, but in skin cells, in treatment of the wounds. And she has already 2 years of the results. And to study for her absolutely new topic, she will not be able time to generate enough results. I decided let's do something connected to the central nervous system, but also connected to what she did. And we decided to do some pilot experiment with nitric oxide synthase, and we get very interesting results. I guess so. I started this project, you can say, mostly from kind of organizational, political reasons, but the results were very, very cute. And I definitely decided to continue to this direction. We administrate agmatine acutely, and we have very clear and dose-dependent increase in the firing activity of the neurons after acute agmatine administration. We also did chronic agmatine administration for 14 days. And here before we have opposite effect of acute and chronic, here results are similar. It's stimulatory effect after acute, it's stimulatory after chronic. So there is no adaptive processes. And what was extremely interesting, which we still don't know how to interpret, we treat rats with agmatine for 14 days. and then tried to check acute effect of escitalopram and VEI. VEI, you remember, it's an antagonist of 5-HT1A receptor. This is a basal activity taken as a 100%. After escitalopram, we get similar decrease in both groups of animals. In the control vehicle and agmatin treated. After administration of VEI, we get recovery of the firing activity, partial recovery in the control animals, but in agmatin treated animals, we get not only recovery, we get even further decrease. And we really don't know how to explain it. We have some hypotheses, but still don't know. Because if a desensitization, let's say that 5-HT1A receptor desensitized, they will be desensitized, we will not see effect of escitalopram. If they don't desensitize, why we don't see recovery after VEI? If some of you would like to choose this topic, you can see the papers on this topic. If some of you will provide us in the project work good explanation, I can take this person or persons to the co-author and we will write paper. What my PhD student that I spoke about, Hande Oshbasak, she's from Turkey, she found very good paper, very interesting paper, which maybe explain our results, but we still don't know how it explain. This is a Chinese group. They worked also with a nitric oxide synthase and depression, and they get published in Science. It says something to be published in Science. What they said, very interesting. What is wrong with our way of thinking when we speak about neurons? We see neuron as a one unit, like one basic functional unit of neural system is neuron. And let's say you check expression of 5-HT1A autoreceptor or serotonin transport in dorsal RFA nucleus of depressed and not depressed subjects, and you see no difference, same expression of these molecules. You say, okay, that probably this molecule is not involved, but it's not true. It's not enough to be them expressed or not expressed. It's a question of how they localize in the cell membrane. What happens if serotonin transporter and 5-HT1A receptor, they closely localized? Because then this area of the receptor will be much less serotonin, extracellular serotonin, because it's immediately soaked into the neuron, and this receptor will be much less activated. And what's happening if they not colocalize on the cell membrane? Then you may have situations that you have with the same same expression, same level, same density of the SERPs and of the 5-HT1A receptors, you'll get more higher activation. And the authors in this say they check the hypothesis that nitric oxide synthase actually mediate this not dimerization, but this interaction between SERP and 5-HT1A receptor. And what they did, it's a very complex study. They did a conditional knockout of the serotonin transporter. Sorry, they did conditional, it's my mistake. They did conditional knockout of nitric oxide synthase. They found this is that it's normal rat mice, so they use mice. NOS and serotonin transporter, they colocalized in neurons, and they interact each with another after the knockout, of course, you don't see anymore. You don't see anymore NOS, but you see only the CERT. What they also found after a NOS knockout, they have antidepressant-like effect in a suppression test and forced swim test, and was very rapid effect. They also synthesize molecule, special molecule Sakura-6, which is doing opposite. It's built together. It's connect together NOS and CERT, and they have depressogenic effect, anxiogenic and depressogenic effect. They also create very heavy study with a lot of experiment, with a lot of very high-tech molecular work. It's a depressogenic study because when you read it and you realize that you cannot do such a quality of research, it's a little bit depressogenic on the researchers. They created at 15C CERT. It's a peptide which mimics the last 15 amino acids of the C-terminal of serotonin receptor, and dimerized with a NOS instead of CERT, and this decreases the CERT-NOS association. And they also found that this treatment with this at CERT-15C first, it's increased serotonergic neuronal firing activity, and second, it has pretty strong and very rapid antidepressant-like effect. They also did simultaneous dissociation between NOS and CERT in the dorsal raphe nucleus, in cortex, and in hippocampus. And they found when you do CERT-NOS dissociation in the prefrontal cortex, it diminishes antidepressant-like effect of CERT-NOS dissociation in the dorsal raphe nucleus, but in hippocampus it doesn't have any effect. This brings us back to this picture that we saw about interaction between dorsal raphe nucleus and prefrontal cortex, which also involve 5C1A receptor, and we believe that this unique effect of NOS inhibition by agmatine, why it still allows effect of serotonin, but does not allow the effect of why. We believe that the change may be happen in cortex and not to the raphe nucleus. As I told, I recommend you to read it, and somebody chooses topics and provide us good explanation, help us to understand it. You can be welcome to the big altar. I think we can finish by this point. You want today to do it? Yes, of course. You have it on the disk. So we will upload it and we will continue it. We will record it also, if you don't mind. Yes.
No, to tell you that's the best presentation I've ever done. No, it'll be fine. In a hectic form. So it was very fun. Yeah. Link of content. There we go. No.
Yes. Kaya, do you see the new presentation?
Yes, yes, I see it.
Okay, very good. Okay, we have what is called in Israel Avirat Sofa course.
Yeah, exactly.
Atmosphere distance breaking. Now you are here and I will.
Okay, so
What we're gonna discuss now is ROC inhibitors or ROC inhibitors as efficient treatment for psychiatric disorders. This is also what I am currently researching in my PhD. What are small GTPAs and the actual ROC family of proteins? So small GTPAs or small G proteins are enzymes that bind GTP. GTP is one of the energy currency of cells. What they do, they're molecular switches that are in charge of many different aspects of the cell life cycle. And the ROC family, which includes RO ABC, ROC 1, 2, and 3, and CDC 242, are considered one of the bigger groups in the more widely researched in the small GTP community in the ROS superfamily. And what they're in charge of is mostly actin dynamics. Actin is one of the main ways that cells move and build themselves. They're one of the main cornerstones of the cell structure, cell skeleton, and they're connected in the CNS to neural outgrowth and neural migration. What is ROCinase? ROC is one of the main effectors of the ROC family. It's mostly in charge of quite a lot of things in the cell, but on movement of cells, development, transmission, connection, morphology, a whole lot, really lots of stuff. In the central nervous system, they regulate damage. They're very much closely related to traumatic brain injuries and other inflammation, oxidative stress. And if they work too much, they actually lead to more problems than they should. They've been detected in overexpression in both Alzheimer's, Parkinson's, and multiple sclerosis. And we know that there is acting issues in quite a few neuropsychiatric disorders, schizophrenia, trauma, depression, Alzheimer's. The and acting in the central nervous system is also deeply connected to cellular signaling. The way that both exosomes, what they call vesicles, and the actual synapse themselves are all built around the acting infrastructure, and as such, they are massively important. Any damage to them causes damage to the central nervous system. Furthermore, we now know from recent studies that the aspect of myelination, of creating white matter, is also deeply connected to actin depolymerization, meaning that the only way that a myelinating oligodendrocyte can actually let go of an axon is by letting go of the actin cytoskeleton. So if we know all of that, and we know that ROC itself is very much connected to actin infrastructure, maybe if we try to target it, we can help retain some of the functionality in psychiatric disorders. And this has actually been not heavily researched, but some of it has already been researched, mostly in a couple of molecules. Today, we're going to talk about two of them. We're going to talk about phosphodiol and about one that you probably know. We'll get to it. So what is phosphodiol? Phosphodiol is not an FDA approved, but an approved substance. It's mostly used for ischemic brain strokes. It helps elevate some of the problems in the vesicular system of the brain, in the blood-brain connection, stuff like that. It has been shown to improve both cognitive decline in stroke patients, and in recent studies, it has been shown to improve both memory in Alzheimer and novel object recognition in Parkinson's, which is really interesting. It's also very much connected to alpha-synuclein aggregation, which is the main issue in Parkinson's disease. Mostly from what we know, the main way of effect of phosphodiesterone to actually interact with anything brain-related is for ROCK inhibition. There's a whole discussion about what actually happens there, which we will talk about in quite a few seconds. It's actually connected to NOS as well. And the other molecule that we're going to talk about is ibuprofen, which I'm sure every single person here has taken at some point. If you've ever taken Advil, you've taken ibuprofen. Ibuprofen is a non-steroid anti-inflammatory drug, very common, FDA approved since the 80s, pretty much been heavily researched, and as such, now we're trying to give it a new life in different disorders and different problems. It affects many different cellular pathways. One of them is ROCK inhibition. More than ROCK inhibition, it's also actually connected to the upstream of Rho A itself, which makes it even more interesting considering it doesn't actually need to invalidate ROCK as a whole. PPAR gamma activation has been suggested as the molecular mechanism. It's connected mostly to how cells divide. It reduced neuroinflammation, which makes sense considering it's an anti-inflammatory drug. But more than that, it has actually been shown to increase myelination after traumatic brain injury. And it also shows better division of neuronal cells. As I said earlier, ibuprofen is considered pretty much safe to use. There's also now ways to make it more brain penetrative and more and better specifically targeting neurons and oligodendrocytes. So it's a good option as let's say as a good molecular target for further research. So how do they actually work? Something we actually saw this this just a minute ago. The main way we believe these molecules are affecting the brain is through actin structures, which are very easily researched. There are a whole bunch of antibodies for fibrous actin. And more than that, there is a substance called phalloidin, which is a chemical toxin derived from fungus that gives amazing colorization of actin structures. I actually do that in my lab. Another thing is that we now know that both phosphodiesterone affects eNOS and ibuprofen affects nNOS, which may mean that the improvements that we see are derived from this nitric oxide synthase interactions. We know for a fact that too much nitric oxide synthase is also toxic to the brain, and once there is an injury or an illness to the brain, it usually rises. Of course, we can use different animal models for different things. We use autistic mice to check if this can actually improve. And we can also tag the substances themselves. There is a way to actually add a fluorescent tag to ibuprofen and phosphodiesterone to actually see what's the interconnected connections to which parts of the cell they actually connect inside the brain, which is amazing. And I just want to show you how you can actually live image the cells with the actin structures. What you see here is a neuronal cell that is moving around through fibrous actin structure themselves. Usually what happens is that you have row A on one side and rock 1 on the other side, and the difference in level causes the migration of neurons. To sum up, we also know there are a whole bunch of other ROCK inhibitors. ROCK inhibitors are mostly used for heart problems. They are known to affect the vesicular system, but are now slowly being adapted for brain and neuropsychiatric issues. The main problem is that they're not specific enough. We know there are 2 ROCK isoforms. There's a whole bunch of because cellular pathways are so complex. Every single thing you take down also causes a whole bunch of stuff downstream that we're not sure about. So we're still trying to make them more specific, both cell-wise and isoform-wise. Further research into actin properties in neuropsychiatric disorders, be it Alzheimer's, depression, PTSD, etc., has been growing in recent years, mostly because there's better ways to live image the actual paths of cells. The main ways we do it now is super resolution. There's also expansion microscopy. So it's easier to actually look at the fibrous actin as a whole. And the main point is that by using clinically approved drugs, we cut down on both the research time and the approval time, obviously. Any questions?
I have one.
Yes.
Actin, as far as I know, but it's very important, the nitric spikes.
Yes. There's also for instance, in my model, there's that we're now trying to see if this actually helps. There are in quite a few models, there is problems with the dendritic spine morphology. In autism, I know that in some models of PTSD, there has been shown that there is problems in development of the dendritic spines. And schizophrenia definitely has problems in the dendritic spines. So it's another avenue of the research.
It was very nice presentation. It was very nice that also the invitations that were announced about the figure.
I think everybody's trying to make a miracle drug, and in the end, it doesn't work. So there's always
For the to upload the copy of your presentation, sure, the website and leave it for more people copy it. Okay, I really recommend you tomorrow, next Sunday. No, you can also even later to upload it. Who wish to do it, virtual presentation, but still, I really recommend you. I think we have pretty friendly forum, so you don't need to be scary about як це ніхера public fair. I think we have nice atmosphere, knowing us for 2 months, so please don't be shy. Next time I really would like my presentation will be pretty short. We will have plenty of time, so please bring your presentation next time.